Hello students, welcome to NEET World. This is Narayani, I am your botany educator. In previous class, we discussed the glycolysis and Krebs cycle, and some part of the Krebs cycle is left out, so we will discuss about today that. So before I am going to start this session, I would like to announce very important news for you here. So here we have started the long term courses for both BPC and also MPC students. We are giving coaching for NEET, EAP set, JEE mains for MPC people and EAP set. So now I am particularly explaining about for the BPC students, that particular concerned lecturers will tell you about them for MPC students. So what are the benefits you get by joining this long term course? So you will get the live plus recording sessions, and also well crafted study material is provided, and also one-on-one mentorship is provided. So it is just for coming just rupees 3,333. This is the online program we are giving just per month 3,333. It is a golden opportunity for you all who are see them as themselves as the doctors. If you are really passionate, this is the best platform for you. If you have any further queries, please contact this number. This is what about our NEET long term course, and it is quite benefit for the 11th and 12th standards as well as the droppers and also repeaters. For the 11th and 12th, if you started the parallelly the coaching here, you will get the good rank in the upcoming competitive examinations of NEET and EAP set. So what are you waiting for? Join with us. So before I am starting the session, I would like to announce another important news for you. So this is our Telegram group. So by scanning this QR code, you get access to our Telegram group. So what are the benefits here you get? So daily quizzes we are providing for biology, chemistry, physics and also mathematics. Live classes, pre-live class schedules will be dropped there. So free resources we are providing, and also whatever you have doubt, you can post it right there. So they're about that particular lecturers will concern that clear you all the doubts for you. So this is one of the best opportunity for you to contact with us. So further any queries, please let me know this by this number. Okay, so let's see the today's session. So we are completed, before I am starting the session, so we completed the we discussed about the cycle, right? So this is the Krebs cycle we discussed about, right? So before discussing about the Krebs cycle, so let's fastly skim that what we discussed in the previous class. So after the glycolysis, the end product of the glycolysis, what is students? That is pyruvate, right? That is pyruvate. The end product of the glycolysis is pyruvate. It is changing into acetyl-CoA in the which organelle? Mitochondria. In the mitochondria, right? So this is the also called as link reaction. For all aerobic organisms, this will be done, right? For one glucose molecule, how many ATP molecules will come? That is 38 ATP molecules will be produced, right? So for one glucose molecule, 38 ATP molecules are produced. For in glycolysis, how many ATP molecules are produced? The gross is 8. Gross is 8 ATP molecules are produced. So remaining 30 will be produced in the Krebs cycle, right? So whatever the end product of the glycolysis is turning into the acetyl-CoA in the mitochondria, that reaction is called link reaction or oxidative decarboxylation, right? So pyruvate is a 3-carbon compound and acetyl-CoA is the 2-carbon compound. So there is a elimination of 1 carbon dioxide is occur here, right? So that is why decarboxylation, oxidative decarboxylation. What is meant by decarboxylation? Decarboxylation mean removal of carbon dioxide. Removal of carbon dioxide, right? So here that is why this link reaction we also called as oxidative decarboxylation. Here the 1 carbon dioxide is going out, and as it is a condensation process, coenzyme A is added, that is why it is the acetyl-CoA. By the end of the glycolysis, how many pyruvates came? There are 2 pyruvates came, right? So here it is also producing 2 pyruvates, sorry 2 acetyl CoA. So 2 pyruvates getting converted into acetyl CoA as 2, and it is the 3 carbon compound, and acetyl CoA is the 2 carbon compound, which means elimination of 1 carbon dioxide occurs here. That is why this process is called oxidative decarboxylation. Right, here the as the enzyme catalyzed here is pyruvate dehydrogenase. Pyruvate dehydrogenase. Whenever the dehydrogenase enzyme came, there is a production of reducing equivalent. That reducing equivalent is NADH. Okay. So we discussed this and we explained about the cycle also. That pyruvate, why it is only getting converted to the acetyl CoA in the only, why it is coming to the only mitochondria, because the enzyme named pyruvate dehydrogenase enzyme is present in the mitochondria. That is why for all aerobic organisms so this process will be occur. For the anaerobic organisms, the strictly glycolysis they depend for the energy purpose, right. So this pyruvate, the end product of the glycolysis, is getting converted into acetyl CoA in the mitochondria, right. So how many pyruvate molecules are produced in the glycolysis? 2, right. So here the 2 molecules of acetyl CoA is produced in the mitochondria, right. So it is a 3 carbon compound, and it here is the acetyl CoA is a 2 carbon compound. Okay. So here it is the 2 carbon compound. So this first we will calculate about the 1 acetyl CoA. So as I said before also, acetyl CoA, why it is only converting in the mitochondrial membrane? Because of the pyruvate dehydrogenase enzyme. In the mitochondrial matrix only the pyruvate dehydrogenase enzyme is present. That is why the pyruvate is getting converted to acetyl CoA in the acetyl CoA in the mitochondria. So that acetyl CoA combines with the oxaloacetic acid. Oxaloacetic acid is the 4 carbon compound. So acetyl CoA is a 2 carbon compound, and oxaloacetic acid is a 4 carbon compound. 4 plus 2, it's 6. Here the producing the compound is citric acid. That is why the first stable product is produced in the Krebs cycle is the citric acid. That is why Krebs cycle is also called as citric acid cycle. And this citric acid cycle having the carboxylic groups, that is why it is also called as tricarboxylic acid cycle also. We called it as Krebs cycle. Why we call Krebs cycle? On the memory of the scientist named Hans Krebs, we call them, it is also called as Krebs cycle. Got it? So this citric acid cycle, this citric acid getting converted into cis-aconadic acid with the help of the enzyme name called aconitase. Right? So for you simple remember what I said to you students. So for the 4 corners there is a production of reducing equivalents, right. There is a 4 corners production of reducing equivalents. What I said to remember, how you have to remember? OCCIO alpha with along S square, they went to listen the FM radio. And at the 4 corners there is a production of reducing equivalents, right. So here see, while converting the malic acid into oxaloacetic acid, there is a production of NAD plus. When NADH, right, NAD plus is oxidized, it is getting converted into NADH, which means here is the enzyme name, what will come? Malic dehydrogenase enzyme, right. Here is also like that only. Isocitric acid getting converted into oxalosuccinic acid. While converting oxalosuccinic acid, there is a production of reducing equivalent. So compulsory there is an enzyme name called isocitric dehydrogenase enzyme will be appear. So coming to here, one of the most important compound, alpha ketoglutric acid. Among the Krebs cycle, only one compound which is having the 5 carbon compound is alpha ketogluteric acid. Here while oxalosuccinic acid is converted into alpha ketogluteric acid, the enzyme acts here, it is the alpha ketogluteric dehydrogenase enzyme. So here is the third corner. When the succinic acid getting converted into fumaric, the enzyme acts here, it is the succinic dehydrogenase. Remember like this for your understanding, the four corners are producing the reducing equivalents. So the four corners are producing the reducing equivalents. Out of isocitric acid, alpha ketogluteric acid, malic while malic acid is getting converted into oxaloacetic acid, here it is also producing the NADH. Only but here succinic acid, when it is converted into fumaric acid, there is a production of FAD plus. Why here only the FAD plus is FADH₂ is producing, why not NADH? Because this having the succinic acid having the binding site for the FAD plus. Okay, that is why here is the reducing equivalent is FAD plus. Okay, so reducing equivalent is FADH₂. Okay, these are the NADH and FADH₂. There are the ATP, they are going to convert ATP only. For 1 NADH equals to 3 ATP. For 1 FADH₂ equals to 2 ATP molecules. Right? So we discussed about this cycle. Let's learn the steps now. So see here. 1 minute. Okay, see here. Krebs cycle. They are asking about to explain the Krebs cycle. So let's see fast. Krebs cycle is a cyclic process. What is meant by cyclic process? The point where it starts, again it's end to that point only. That is why Krebs cycle is a cyclic process. So first the Krebs cycle starts from when the oxaloacetic acid combines with the acetyl-CoA forming the citric acid. Again it getting back to the same position. That is why it is a cyclic process, which occurs in all aerobic organisms. Aerobic organisms means which are having depending on the oxygen. It happens, Krebs cycle happens all aerobic organisms to generate energy. It takes place in this which organelle? Mitochondria. That is why mitochondria is, we call it as a, from 10th standard onwards we are learning about that, right? Mitochondria is a powerhouse of the cell. So in Krebs cycle, acetyl coenzyme A is oxidized to form carbon dioxide. Here is the, when the acetyl coenzyme A is producing, that time decarboxylation is happening, right? As it is a condensation process, water is also eliminating. One carbon dioxide is also eliminating there and hydrogen is also eliminating there. So also ADP is converted into energy rich ADP. So why this all Krebs cycle is happening, glycolysis is happening to produce energy. So what is the currency form of the cell? It is a ATP. So the energy will will name as the ATP, adenosine triphosphate. Right? For one glucose molecule, how many ATP molecules will produce? 38 ATP molecules will produce. First that glucose molecule enters into the cell. Right? So first the first reaction is glycolysis, it produces the 2 pyruvates, right, with the series of steps. After this, these 2 pyruvates enter into the mitochondria organelle, right, mitochondrial matrix. Why it is only goes for the mitochondrial matrix? Because the enzyme named pyruvate dehydrogenase enzyme is present in the mitochondrial matrix. There itself occurs the Krebs cycle process, then it produces the energy, right? So whatever we eat the food contain carbohydrates, right? Proteins, fats, right? So ultimately carbohydrates are having the sources like what are the sources of carbohydrates? Nothing but glucose, fructose, right? These are all the carbohydrate sources. When we are eating, it is in the form of polysaccharides. So we are, while we crushing by the digestion, so it becomes the simple molecules, right? So after the first the digestion starts at the mouth. and it is completed in the stomach, right? While crushing the food, it starts in the mouth. That polysaccharides become the simple monosaccharides that form is glucose, fructose, like that. So proteins also are, they are also converting into the simple molecules. So ultimately these carbohydrates, proteins, amino acids, they are ultimately getting converted into the acetyl-CoA to produce the energy. This is ultimately getting converted into acetyl-CoA. If they ask what are the key intermediate molecule of carbohydrates, proteins and fats, it is a acetyl-CoA is the key intermediate molecule. When it is turned into the acetyl-CoA, it gives to the energy. So it enters into the Krebs cycle, then it forms the energy, right? So in our body, almost they are having 3.72 trillion cells are having. So in all cells there should be a process of glycolysis. Krebs cycle will be occur to generate the energy, right? So see here, Krebs cycle reactions. To understand this steps is also very required for your examination. So as it is the IPE, you have to elaborate the answer. So in section C, they will give 3 questions, you have to write 2. So almost they give the glycolysis, Krebs cycle. Among these 2, you will definitely get 1 for sure. Maximum you will get 1 for sure. Other than this, you will get from the photosynthesis in respiration in plants. So in respiration plants, these 2 are very important. Very important. You have to study it. So you have to understand the concept, then it will be very easy for you. So out of these 2, you will get definitely 1. So as it is the essay question, you have to elaborate the answer. You have to write, so where it is happening, what is the process it is, and steps. So series of steps you have to write it. So the first acetyl-CoA is the link reaction. So in the link reaction, what is produced? Acetyl-CoA is produced. So 2 pyruvate molecules at the end of the glycolysis, what are the pyruvate is produced, it is getting converted into the acetyl-CoA in the mitochondrial matrix. That acetyl-CoA combines with oxaloacetic acid, which is the 4-carbon compound. Acetyl-CoA is the 2-carbon compound. Okay. So by combining with acetyl-CoA, how much it is became? 4 plus 2, 6. So 6 carbon compound named citric acid by the help of the enzyme name called citric synthase. So as it is the coenzyme A, here it is the eliminating. So as is the first process, we call it as condensation. As it is eliminating, condensation. When the coenzyme is coming or going out, that process we call it as condensation. Okay. Acetyl-CoA combines with oxaloacetic acid along with the water, it forming the citric acid by the catalyzed enzyme name called citric synthase. What is meant by catalyzation? What is meant by catalyzation? It is a fast up the reaction. So enzymes, why the enzymes are acting? To fast up the reactions. Without enzymes, we cannot imagine any reactions, because enzymes makes process easy. So if without enzymes, it will take 2 days. With enzymes, it will take 1 day. Okay, just like if you are in a school, you have to go home. By walk, it takes time. By bus, it takes less time. Like that only enzymes also. If you have the enzymes are acting, it will take the less process. If the enzymes are not acting, it will take more process. That is why the catalyzation of enzymes are very important. Okay. So here see the next process. After citric acid is combines with the, so here is the process is dehydration, which is elimination of water. So here is the first one, water is came in. So now it is getting out the water is, there is a loss of water. So that is what dehydration. For the citric acid, in the presence of enzyme called aconitase, it forming the cis-aconitic acid. So here is the elimination of water molecule. That is why this process name called dehydration. So, I will give the mnemonic also for you. First I will explain the steps, then we will see the mnemonic. So, next process, first condensation. In the condensation what is happened? There is a elimination of coenzyme A is occurred. So, here it is a 2 carbon compound. So, oxaloacetic acid is a 4 carbon compound. The 4 plus 2 is 6 carbon compound is produced, that is citric acid, in the presence of enzyme name called citric synthase, right. So, the next process dehydration. In the dehydration, what is meant by dehydration? Elimination of water molecule from the citric acid by the enzyme name called aconitase. The elimination of the water is occurred. When it is elimination occurred, the citric acid became the cis-aconitic acid. So, see here, third step is hydration. What is mean by hydration? Adding of water molecule, right. So, adding of water molecule is called hydration, right. Dehydration means taking out of the water molecule. Here is what is happening? Adding of the water molecule is happening, for the which the last compound is cis-aconitic acid. So, here is the water is added. Okay. So, with the help of the enzyme name called aconitase, it become the isocitric acid. So, cis-aconitic acid is also 6 carbon compound, citric acid is also 6 carbon compound, isocitric acid is also 6 carbon compound. Okay. So, here is the hydration step is completed. Next, oxidation. Here it is the oxidation, which means here is the process oxidation. So, whatever the isocitric acid is the 6 carbon compound. So, adding with the NAD plus oxidized form. Whenever the NAD plus is came, there is a formation of NADH, which means the enzyme name what will come here? Dehydrogenase, right. Isocitric dehydrogenase enzyme added to produce the oxalosuccinic acid. Oxalosuccinic acid, this is also 6 carbon compound. As it is the having the binding site for NAD plus, it is produced the NADH plus H plus. Okay. So, with the help of the enzyme name called isocitric dehydrogenase enzyme. So, now this process is called oxidation. What we call oxidation. Coming to the next one, decarboxylation. What is meant by decarboxylation? Elimination of the carbon dioxide here. So, here the oxalosuccinic acid is a 6 carbon compound. Here it is a 6 carbon compound, right. So, in the presence of enzyme decarboxylase, oxalosuccinic decarboxylase enzyme producing the alpha ketogluteric acid. It is the 5 carbon compound. Why it is 5 carbon compound? Here is the elimination of what occurred? Carbon dioxide is occurred, right. Here it is a carbon dioxide. There, as it is the elimination of carbon dioxide is occurred, so it is become the 6 carbon compound become the 5 carbon compound. Okay. Coming to the next step, oxidation 2. Here it is also what is producing? Reducing equivalent is producing. Alpha ketogluteric acid is the 5 carbon compound. Wait. So, 5 carbon compound. One minute. So, alpha ketogluteric acid is the 5 carbon compound. 5 carbon compound is by the action of the enzyme name called alpha ketogluteric dehydrogenase enzyme, it is forming the succinyl CoA. There is the action of coenzyme A also here and NAD plus also here. NAD plus produced NADH, right. So, here is also elimination of carbon dioxide is occur. See, here is a carbon dioxide is occur, elimination of the carbon. It is a 5 carbon compound, right. Then the succinyl CoA is the 4 carbon compound. Got it? Because the elimination of the carbon dioxide is occur by the help of the enzyme name called alpha ketogluteric dehydrogenase enzyme. With the help of this enzyme, the 5 carbon compound become the 4 carbon compound. Here that name is succinyl CoA. Here is addition of coenzyme A is also happening. It happens here, right. So, this is how the 6th step is done. Now, see the next steps. Seventh step, it's a cleavage. Cleavage, so here is the substrate level phosphorylation occur. What is meant by substrate level phosphorylation? Which means directly they are producing the ATP. That is nothing but the substrate level phosphorylation. So here see in step 7, cleavage. In this cleavage step, there is succinyl-CoA. This ADP with inorganic phosphate, PA means inorganic phosphate, becomes a succinic acid. This is also 4 carbon compound, this is also 4 carbon compound. There is no elimination of carbon dioxide here, but here is the happening substrate level phosphorylation is occur. So this ADP become the ATP. ADP become the ATP. So here is the elimination of succinyl-CoA, that is why it form the only succinic acid. This succinic acid in the step 8, as it is a 4 carbon compound by the dehydrogenase enzyme. So here is FAD plus oxidized form is reduced into FADH₂. So this oxidized form forms the reduced form, that is FADH₂. So with the help of the enzyme name called succinic dehydrogenase. Succinic dehydrogenase. Okay. So with the help of that enzyme, it produces the reducing equivalent name called FADH₂. Why not NADH? Because this succinic acid having the binding site for FAD plus. That is why here the formation of FADH₂ occurred. Right? Coming to the next step. Next step is hydration. What is meant by hydration? So here is the adding of water molecule. Dehydration means elimination of water molecule. Here is the fumaric acid. So whatever the fumaric acid is also 4 carbon compound. This 4 carbon compound adding with water with the help of the enzyme fumarase, nothing but the isomerase compound, it forming the malic acid. This malic acid in the 10th step, oxidation step 4, right? So here it is what is forming malic acid combines with the NAD plus, it forms the with the malic dehydrogenase enzyme, it is forming the oxaloacetic acid. So where we started the process, again we came back to that place, right? That is why it is a cyclic process. So malic acid, oxaloacetic acid is also 4 carbon compound. With the help of the enzyme, we turned into oxaloacetic acid is malic dehydrogenase enzyme. This NAD plus oxidized form the NAD plus reduced form. NADH plus H plus in the reduced form. Here the steps have to, this is how the steps are occurred in the Krebs cycle. So this is what about the steps. See, now I will give the mnemonic for you to to remember easily in the examination. So CD, Hodo, Coho. CD, 1 minute. Okay. CD, Hodo and Coho. CD, Hodo, Coho. See, first I will write here, then you will understand. So remember like that, then it will be get easy for you. CD. Then next, 1 minute. Okay. CD, Hodo and Coho. Wait 1 minute. CD, Coho, 1 minute. CD, Hodo and Coho. Remember the step names like this. CD means condensation, D means dehydration. Like that you remember the steps names. Then it will be quite easy for you for your examination. Okay, the steps are very important for you. You have to write the steps. Then it will be you will get the marks 8 out of 8. Right? So see here. So now let's see if you can't draw this this cycle. So then prefer this cycle. So this is what happens. So this is the cycle what happens exactly in the Krebs cycle. So that pyruvate is getting converted into acetyl-CoA, then acetyl-CoA combines with oxaloacetic acid, then oxaloacetic acid forms the citric acid. So oxaloacetic acid is the 4 carbon compound, acetyl coenzyme A is the 2 carbon compound, it forms the citric acid is the 6 carbon compound. Then it is again formed the cis aconitike acid with the help of the enzyme name called aconitase. Then again with the same enzyme it forms the isocitric acid. Then it is forming the oxalosuccinic acid. So while forming the isocitric to oxalosuccinic acid, there is a reducing equivalent named NADH plus. H plus is formed with the help of the enzyme name called isocitric dehydrogenase enzyme. This oxalosuccinic acid getting converted into the alpha ketogluteric acid with the help of the enzyme name called oxalosuccinic decarboxylase, which means elimination of the carbon dioxide is occurred, right? So here is the it is the 6 carbon compound, now it become the 5 carbon compound. The only 5 carbon compound in the Krebs cycle is that is alpha ketogluteric acid. This alpha ketogluteric acid getting converted into the succinyl CoA, the enzyme name called alpha ketogluteric dehydrogenase enzyme. Then this succinyl CoA forms the substrate level phosphorylation occur here, forming the succinic acid. There is a formation of GTP in the animals, ATP in the plants, right? GTP is nothing but ATP only, okay. So this succinic acid getting converted into fumaric acid in the presence of enzyme name called succinic dehydrogenase. Whenever that succinic dehydrogenase enzyme came, there is a formation of reducing equivalent. Which reducing equivalent here? Not NADH, it is the FADH₂. Why FADH₂? Because succinic acid having the binding site for FAD plus, right? So it is reduced into FADH₂. This fumaric acid getting converted into the malic acid, nothing but isomerase compound only, malic acid also. Here is with the help of enzyme name called fumarase, there is a addition of water, with the help of addition of water. Then this malic acid getting converted into oxaloacetic acid in the presence of an enzyme name called malic dehydrogenase. Whenever the dehydrogenase enzyme came, there is a production of reducing equivalent. That reducing equivalent is NADH₂. Okay, this is what about the Krebs cycle. Let's see if you can't, if you take a screenshot of it, if you are not able to write all these steps, so it is idealized to write all these steps, then it will get very easy for you. What is happening in the Krebs cycle, you know about very clearly. So if you can't, if you are not able to draw this cycle, this is the simple cycle you can draw in your examination. But if you write that cycle, it is very more effective, right? And so let's see how many NADH are produced. Let's see here. In the glycolysis, first make a list of it. In the glycolysis, how many ATP are produced? So in glycolysis, how many ATP are produced? How many NADH are produced? We'll see. So in how many NADH are produced? 2 NADH are produced, right? Okay. 2 NADH are produced. How many ATP are produced? 2, right? Because what in an energy requiring phase we utilize 2 now. So then we in the 6 to 10 step 4 ATP are produced directly. Substrate level phosphorylation occurred, 4 ATP are produced. But so as we utilized in the first 5 steps, we are eliminated that 4 minus 2, 2. So here is the 2 ATP are produced. Total how many there is any production of FADH₂? Any production of FADH₂? No, right? There is no production of FADH₂. So here is the total is 8 because 2 NADH, 2 into 3. So 1 NADH is equal to 3 ATP, right? So 1 FADH is equal to 2 ATP, which means what happened? So 2 into 3, 6 ATP are produced. So 6 plus 2, 8 ATP are produced in the glycolysis. Now see the link reaction. In the link reaction oxidative decarboxylation, how many NADH are produced? So end of the glycolysis 2 pyruvates are produced, right? So for the 2 pyruvates we have to include. So there is for one acetyl CoA to produce from the pyruvic acid, there is a production of NADH, right? So here is the, further we want for 2 pyruvates, right? So which means for 1 pyruvate, 1 acetyl-CoA. For 2 pyruvates, 2 acetyl-CoA are produced, which means the reducing equivalents are produced here are 2 NADH. 2 NADH. There is no ATP is produced here, there is no FADH is produced here, which means 2 into 3, 2 into 3, 6 ATP are produced, right, in the Krebs cycle. In the Krebs cycle, how many reducing equivalents are produced? There are 3 NADH are produced, right? 3 3s are 9. 9 ATP molecules are produced. FAD, is there is any ATP? Yes. For 1 pyruvate, 1 ATP. For 2, for 1 acetyl-CoA, 1 ATP. For 2 acetyl-CoA, 2. Right? 2 ATP is not produced, GTP is produced, right? So in the, okay, ATP is also, here we write, ATP is also nothing but GTP only. Okay, so here is the how many GTP are produced? There is a production of 2 GTP molecules, nothing but ATP only. Okay, for your understanding I will write here like this. Then FADH₂, 2 FADH₂ are produced. 2 FADH₂ are produced. 2 into 2, 4, right? 4 ATP molecules are produced, right? 9 plus 2, 10, 11. 11 plus 4, wait, 1 minute. 15, right? See here, how many? 15. So this is what the energy, high energy produced in the Krebs cycle. 1 minute. 1 minute. 3 ATP are produced now. So 3 NADH are produced now. For 1 cycle, 3 NADH are produced in the Krebs cycle. For for 2 acetyl-CoA, there are 6 NADH are produced. 6 into 3, 18. 6 into 3, 18. Right? Okay, now see the calculation. 18 plus 2, 20, 24 ATP are produced in the Krebs cycle. Total how much? There is a production of 38 ATP molecules are produced in the, for 1 cellular respiration, right? But if it is a gross, which means total energy is produced is 38 molecules of ATP, but for the net it is we have to consider the 36 only, because the 2 are utilized in the electron transport chain. So when we are discussing that I will say you, but if they ask the total how many ATP molecules are produced, total 1 minute, okay, total 38 ATP molecules are produced. 8 from the glycolysis, in link reaction it is 6, and in Krebs cycle it is 24. So total 38 ATP molecules are produced in the cellular respiration. If they ask the what is the count of the ATP molecules in the cellular respiration, it is 38 ATP molecules are produced in the cellular respiration. So if they ask net, that is 36 only, because 2 are utilized in the electron transport chain. Okay, so don't confuse it. So I will say in the next class also, in a NEET table, then you will get quite efficiently you can understand. This is about the Krebs cycle. So for 1 glucose molecule, to generate the energy, the glycolysis, link reaction and also Krebs cycle have to be done. Then it will get the energy. Total how many for 1 glucose molecule it produces the 38 ATP molecules of energy is produced for 1 glucose molecule. From the carbohydrates, proteins, fats, the key intermediate molecule is acetyl-CoA. Okay, so we will discuss this balance sheet in the next class. So I hope you all clear about this today's session. So let's see in the next session. So before I am winding up the class, I would like to solve some of questions right here, then it will get easy for you. So in animal cells, see the first question here. In animal cells like muscles, when oxygen is inadequate, which means oxygen is less in animal cells, for cellular respiration during exercise, when I said the glycolysis, when the vigorous exercise is done by the any person or in the animals, what will happen? There is a pyruvic acid is reduced into lactic acid with the presence of enzyme name called they are asking. So by the enzyme they are asking, which means which enzyme? This pyruvic acid turns into lactic acid in the animals when there is a insufficient of oxygen. Which enzyme that is? Lactate dehydrogenase enzyme. With that enzyme, the pyruvic acid is getting converted into lactic acid. Okay. So coming to the next question. In eukaryotes, pyruvate formed by glycolytic metabolism. Glycolytic cycle, glycolysis same. So in eukaryotes, pyruvate formed by glycolytic catabolism. Of course, in the series of steps of glycolysis forms the pyruvate only, right? Of carbohydrates is converted into nothing but carbohydrates. This is the polysaccharide. This polysaccharide is getting converted into the monosaccharide. So first when we eat the food, that is in the polysaccharide form, right? So it will get simple in the digestion. In the complete digestion occurs in the stomach. Then it will become the simple monosaccharide name called glucose, right? So that is what they are saying. Carbohydrates is converting into acetyl CoA, which means nothing but glucose only. Okay, is converted into acetyl CoA by complex set of reactions. By a complex set of reactions catalyzed by pyruvic dehydrogenase in the. They are asking which place they are asking. Is they are asking as it is occurs in the cytosol? No. In the cytosol, the process of glycolysis occurs. They are asking carbohydrates is converted to acetyl CoA. They are asking their formation of acetyl CoA. Where it is the forming of the acetyl CoA in the cell? In the mitochondria, right? Why is only the mitochondria? Because there is a presence of pyruvic dehydrogenase enzyme present in the mitochondrial matrix, right? So here is the direct answer is mitochondrial matrix. Cytoplasm occurs a glycolysis. Then there is no hyaloplasm we discussed here. So mitochondrial matrix is the right answer for this question. Okay, don't confuse. So because if they ask the carbohydrates, so carbohydrates are nothing but the food we eat, right? So when it is becoming the digestion is happening, it became the monosaccharide, nothing but glucose, right? So this is the we have to understand the given question. So how they are asking? If it is not they are asked directly glucose, they are asked the carbohydrates. What is the carbohydrate sources? Glucose and fructose, right? So this is the second question. Let's see the third question. So one molecule of GTP is synthesized in the citric acid cycle during conversion of. So here when the substrate level phosphorylation occurs in the Krebs cycle, in the succinyl CoA, when it is forming the succinic acid, there is a occurring of substrate level phosphorylation is occur. See here. So see, this succinyl CoA, substrate level phosphorylation occurs in the Krebs cycle when the succinyl CoA getting converted into succinic acid. There is a formation of GDP, GDP. There is a formation of GTP. When the GDP is combines with inorganic phosphate, there is a formation of GTP. GTP is nothing but ATP form only. Okay. So they are asking which in animals is wait 1 minute. One molecule of GTP is synthesized in the citric acid cycle during conversion of succinyl CoA to succinic acid. So here it is the direct question, right? So if you know the cycle very well, you can ask some of the questions directly. So the condensation reaction, what is meant by condensation reaction? There is addition of coenzyme A, right, of the tricarboxylic acid cycle is catalyzed by the enzyme. They are asking the condensation reaction happened in the tricarboxylic acid. What is the tricarboxylic acid? It is the citric acid, right? Why we call Krebs cycle is also called a citric acid cycle? Because in the citric acid there is having the tricarboxylic acids. That is why citric acid is also called as tricarboxylic acid cycle. And why it is called citric acid cycle? Because the first stable compound produced in the Krebs cycle is the citric acid, right? So it is catalyzed by the enzyme. They are asking which enzyme is catalyzing. Here it is a direct question, not hexokinase, it comes in the glycolysis, right? Pyruvate dehydrogenase, it comes in the link reaction. So decarboxylation is also nothing, it is not that, because here it is giving the condensation, right? Not decarboxylation, there is no elimination of the carbon dioxide here. Here is the answer is citrate synthase. Citrate synthase. So this is about the 4 questions. Let's see the one more question here. So do this as a homework for you all, then we'll see in the next class. I will explain this question in the next class. Take a screenshot of it and answer in the comment section, or else directly message me, or else drop your answer in the chat box or else in the comment box. Okay, this is about the today's session. So before and winding up the class, I would like to announce very important news for you here. So once again I am reminding you students, if you are really passionate to see yourself as a doctor or else a top engineer in the top institutes, then here is the best platform for you. So here we are providing the online course for just rupees 3,333 per month. Okay, what are the benefits we get you by joining with us? So you will get the live classes plus recording sessions, well crafted study material is provided. If you do the, if you listen the classes, if you do the, strategy plan of us and, do the daily DPPs, daily practice tests, so it is enough for you. So you will definitely crack you will get definitely secure the good rank in the upcoming competitive examinations like NEET and EAP set examinations. So here we are, and one of the most important thing is we are providing the one-on-one mentorship. So if you have any doubt, you can directly contact with us. And we are using the digital panels, so thereby you can understand with animations. So it is quite useful for you. We are providing both offline also and online also. For offline people, you can come to our institute name called NEET World, present in Narayani Ma'am, Hyderabad. If you want to join through online, please contact with this number. If you have any queries also, please let me know by this number. If you are really passionate to become, to see yourself as doctor and in the top engineers in the top institutes, it is the best platform for you. So this is what about that. And one of the very important news here. So this is our Telegram group. So by scanning this QR code, you can access our Telegram group. So while for every competitive examinations, doing numerous mocks are very important. Then it will boost your confidence, right? So we are providing daily quizzes of biology, chemistry, physics and mathematics, and whatever the pre-live class schedules will drop there. So there you can join on time with us. And free resources, whatever the free resources are there will drop right there. And also one-on-one doubt solving sessions will be there. So there we will keep the doubt sessions also, and also if you have any doubt, you can directly post your doubt right there. So thereby we can check it out and clear your doubt. So if you have any queries, please let me know by this number. Okay. So this is what about this today's session. So let's see in the tomorrow session. So don't forget to write the notes. So always prefer to write the notes, then it will be easy for you. As it is the cycles, you have to write the notes. Okay, this is about today's session. Thank you.